Namaskar, welcome to PK's classes. Today we will study the nuclear receptor. These nuclear receptors are basically the intracellular receptors. The other three receptors, ion channel link receptor, G protein coupled receptor, and tyrosine kinase linked receptors are located on the cell membrane, whereas this nuclear receptor is present within the cell, inside the cell, hence they are the intracellular receptors, and some of them are present in the cytoplasm, and when the ligand binds they enter into the nucleus, and some of them, in the absence of the ligands, they are located in the nucleus. So both are nuclear receptors, and we can classify them as cytoplasmic receptors and nuclear receptors. Cytoplasmic receptors located in the cytoplasm, and when the ligand binds to it, they or agonist binds to this receptor, they are translocated into the nucleus. And the second type, they are located inside the nucleus, whether the ligand binds or not bind. So in the absence of agonist also, they are located in the nucleus. And this type cytoplasmic receptor, they are located in the nucleus once the agonist binds to it. So these are the two types. And the cytoplasmic receptors example is glucocorticoid receptor, androgen receptor, mineralocorticoid receptor, progesterone receptor. All are examples of cytoplasmic receptors, which when these agonists like glucocorticoid, say glucocorticoid binds to glucocorticoid receptor, it will enter into the nucleus. And nuclear receptor, they are located in the nucleus. For example, peroxisome proliferation activator receptor, alpha, PPAR, gamma, retinoic acid receptor, thyroid receptor. These are some of the examples of these receptors which are located inside the nucleus. All are examples of nuclear receptors. Now, if you see the structure of a nuclear receptor, it has one DNA binding domain. So we can divide it into three parts. One DNA binding domain, and the other is the ligand binding domain, and the third part is the transcription activating domain, because these are the receptors which cause gene transcription by binding to the specific gene, and the part to which they bind is called the receptor binding element, or that receptor response element, binding element or response element. So when this DNA binding domain binds to the receptor response element in the DNA, there will be transcription of the DNA or gene through the transcription activating domain of the receptor, and DNA will be converted to messenger RNA because of this transcription, and then by translation messenger RNA will produce specific proteins. So this is the inactive nuclear receptor where ligand is not binding, and this is the active nuclear receptor where ligand binds to the receptor, and then there may be, in some cases, there is binding of a co-activator protein to this complex, so that there will be DNA binding with the receptor response elements. DNA binding domain of the receptor will bind to the receptor binding element or receptor response element of the DNA, and there will be gene transcription followed by production of proteins. So let us take one example of a nuclear receptor, PPAR, gamma peroxisome proliferation activated receptor, which is located in the adipocytes, skeletal muscle and liver. So when the agonist thiazolidine diones like rosiglitazone, troglitazone, pioglitazone, they are administered, they will enter into the nucleus and will bind to the PPAR gamma, which forms a dimer with another receptor, retinoic acid receptor, RXR, and this thiazolidine dione and receptor complex will bind to the thiazolidine response element or PPAR response elements present in the DNA, which will regulate the gene transcription. And they are called as insulin sensitizers. They increase the insulin sensitivity. So the thiazolidine diones binds to the PPA response elements, which will cause the adipose cell differentiation. So the pre-adipocytes will be converted to adipocytes, and adipocytes will secrete adiponectin, which increases the expression of PPAR gamma angiopoietin related genes, so which will cause angiogenesis, development of blood vessels. So there will be adipose tissue development, and adipocyte function is to store the free fatty acids as triglycerides. So there will be increase in fatty acid storage in adipocytes as triglycerides, and there will be decrease in the secretion of free fatty acids from triglycerides. So triglyceride storage in adipocytes will be there. And second, adiponectin also activates AMP activated protein kinase, which will phosphorylate and inhibit this key enzyme involved in fatty acid synthesis and beta oxidation, acetyl coenzyme carboxylase ACC. ACC1 and ACC2 are inhibited, so it will inhibit fatty acid synthesis in liver and adipose tissue, promote fatty acid oxidation in skeletal muscle and liver. So all these will result in decrease in free fatty acid in the blood. And when these free fatty acids are uptaken by the liver and skeletal muscle, and similarly glucose is also uptaken by the liver and skeletal muscle. When free fatty acid is more, it will inhibit the glucose uptake. So when the free fatty acid level is decreased by thiazolidinediones by acting as agonist on PPAR gamma, there will be increase in GLUT4, glucose transporter 4 expression, and increase in uptake of glucose into the skeletal muscles and hepatocytes, and that's why we call that will result in decrease in glucose level, and that's why we call the thiazolidinediones as insulin sensitizers. So they increase the insulin sensitivity. They increase the glucose uptake into the liver and skeletal muscle by decreasing the free fatty acid levels. Actually, when there is increase in free fatty acid level, that will inhibit the uptake of glucose. So by decreasing the free fatty acid, we can increase the glucose uptake. And let us take another example of the cytoplasmic receptor, glucocorticoid receptor. Glucocorticoid receptor in its inactive form is bound to heat shock protein HSP, and corticosteroids act as agonist here. Corticosteroids to enter into the cell, they bind with a carrier protein called corticosteroid binding globulin, so that CBG helps entry of corticosteroid into the cell. Then once it comes into the cell, in the cytoplasm, it will bind to the glucocorticoid receptor. So after binding, there will be removal of the heat shock protein, and that will be followed by dimerization of the receptor. So corticosteroid enters into the cytoplasm. The glucocorticoid receptor, which is bound to heat shock protein in its inactive form, when glucocorticoids bind to this receptor, the heat shock proteins are removed, and there will be dimerization of the receptor, and that receptor dimer will bind to the glucocorticoid response elements in the DNA, and that will regulate the gene transcription, DNA to messenger RNA to proteins, required proteins will be formed. So this is all about nuclear receptors. Thank you.